On the global scale of human health, there's one underlying problem that affects us all, and it could have been made more clear than by the recent pandemic. It takes a lot of time, a lot of money to develop new drugs, and that's costing people their lives, jobs, and long-term well-being. So how can we address this problem?
So, take penicillin, a drug that everybody knows that, definitely was not modern drug discovery. It was, just a plate left by accident overnight. And that single plate, allowed to save millions of lives over decades. Imagine that didn't happen. Discovering drugs just by, observing nature and this kind of accidents is clearly very efficient. And so, drug discoveries allows us to systematically search for this kind of drugs and be able to target diseases in a systematic way.
A computer-driven method that helps us understand how drugs interact within our bodies is the use of docking programs. Docking refers to the action of a drug binding to a protein. The goal of a docking program is to find out where and in what conformation a drug binds to a protein. The results generated by docking programs are helpful in discovering a drug's effect. Proteins are the main mechanisms by which life operates. By binding to a protein, a drug can prevent it from carrying out its function. This can be used to fight disease by preventing unwanted functions, like viral ones, from occurring. By knowing where a drug binds to a protein, scientists can change the drug to be more complementary to a protein, thus rendering it more effective.
As computers became widely available, they were increasingly used to predict where a drug docks to a protein. These first computational methods randomly searched a protein for the best docking spot, which was time-consuming and computationally expensive.
Recently, methods like DiffDock, which is an AI model that tackles the problem of docking, have emerged. DiffDock uses a new technique called diffusion and is tasked with finding where a drug docks to a protein when given only the drug's structure and the protein structure.
Well, what we're really going after is somewhat of a different setup compared to traditional docking methods. Traditional docking methods were developed in a relatively restricted computational problem where they excel at, which is, where you have a very specific binding pocket rigid in a protein, and you're trying to find how a molecule would fit. With DiffDock, we are trying to take a first steps in a much larger goal, which is the one of actually being able to understand, how binding of a small molecule and a protein works on a larger scale, so at a whole protein scale, in a setting where the protein might be flexible.
DiffDock learned how to dock a drug and protein by training on thousands of experimentally determined drug-protein structures. As a fusion model, DiffDock is trained by gradually altering the position of the drug in the determined drug-protein complex. Then the model is asked to predict how the data was altered, so it is able to regenerate the correct docking locations. To alter the correct docking, the drug's position, orientation, and intermolecular angles are randomly changed. After applying these changes, DiffDock is asked to reverse the altering. Then the model learns how to improve based on the accuracy of its guess. To predict unknown dockings, the process used to train the model is reversed, starting with multiple copies of the same drug randomly distributed around the protein. At each step, DiffDock predicts a movement for each individual instance of the drug. In the end, we get a map of predictions, with each drug-protein docking being given a score by another model based on its confidence in the docking.
DiffDock allows researchers a faster way to find drug-protein dockings. This can guide researchers into creating hypotheses on how their drugs work, ultimately allowing them to take further steps upon improving their drugs.
One example of where researchers already used DiffDock was at the University of Washington, where they had a bunch of proteins and now they needed a single ligand, and now they needed to find out to which of those proteins does the ligand bind. And for that, they then used our blind docking method to dock the small molecule to each one of them. And then they used some downstream calculations based on that structure prediction to find out to which protein the molecule binds the most strongly.
Because of DiffDock's relative speed, it can be used to predict thousands of potential drug-protein dockings. While previous methods predicted approximately correct dockings 23% of the time, DiffDock maintains 38% with this same metric.
With fast and relatively accurate predictions, DiffDock can be used to assist scientists in creating better drugs. DiffDock unlocks the new capability of docking to computationally generated protein structures, such as structures predicted by AlphaFold-2. Previous investigations found that traditional methods struggle with the slight inaccuracies of computationally generated structures, but since DiffDock mainly uses a simpler representation of the protein, it is still able to perform well. With new AI tools like DiffDock, drug discovery is becoming a more effective and streamlined process, potentially saving people's lives, jobs, and supporting their long-term well-being.
Maybe methods where the computational tool just completely generates the new molecule, the new drug from scratch, they are a bit off, a bit of a ways off. But where we see the much more impact, right, and where the computational methods are doing a job already in drug discovery is in helping drug discovery by making this affinity prediction, by guiding the search, by telling you what sort of experiments you should do. And yeah, there we already see large impacts of computational tools in drug discovery.